Hello, everyone. So we're going to get started. Welcome to today's session in the Microbiome Informatics webinar series. I am Ahmed Zayed. I am going to be leading this session today on the advanced ecological statistics for microbiomes. I am a research scientist here at The Ohio State University. I work with Dr. Matthew Sullivan and Dr. Virginia Rich, and I am just going to give you a very quick background, like introduction about me and what I do, and then we're gonna jump into the session. So I have been studying since I came to OSU. I have been studying microbial ecology and evolution, and I've been also studying gene expression from the omics and meta-omic data sets. So for example, what I'm showing you here on top, this is some of the figures from our most recent paper in Science, where we have been looking at RNA viruses across the global ocean. And we have been trying to understand their taxonomic diversity, as well as their diversity metrics across the global ocean, and trying to link this ecology and this biology to the environmental variables, to the things that we can see, the parallel measurements that we are making across the sampling sites that we had from the global ocean. So this is, here is a snapshot of the kind of like correlations that we were able to construct between RNA viruses and DNA virus diversity and different environmental variables. It looks a little bit more complicated than direct correlations because we used some sort of pre-processed data, and that is basically trying to account for autocorrelation between the environmental, the autocorrelation that happen between different environmental variables that we have in our metadata. So this is something that I used a tool that we're going to discuss today. It's called WGCNA. So this is something that we are going to walk through the code of in today's session. So this is one example for how to use networks in your work to simplify the data, and so that you can be able to extract meaningful information from the huge amount of data that you can collect during your experiment. There are other applications. So we also can, for example, link specific functions to the ecosystem outputs or the feature that you're interested in. So what you're looking down here on the lower left, this is also something that I have participated in. This is a study from mice that we have been looking at the variation in the microbial community composition in a genome-resolved fashion. And this is across different injury levels that happen at the spinal cord of these mice. So basically, you can also leverage some of the multivariate analyses and some of the network analyses so that you can link the specific enrichment of taxa with the disease status that the mice are suffering from. Again, this is something that you can use one of the tools that I'm going to discuss today, the WGCNA for. So the difference that we had here is that we were able to simplify the data to just a tiny set of the different metabolic modules that we were able to link with the enrichment of these metabolic modules, basically in the normal or the control samples versus the animals that are suffering from this spinal cord injury. You can even go beyond that and start to look at genome-resolved mapping of the expression of these genes in the metatranscriptome and the metaproteome, and map that to your kind of genome maps. So there are a lot of things that these kind of tools that we're going to talk about today can help you to do, can simplify things pretty a lot for you, so that you can kind of invest your time on the biology rather than trying to sift through the massive data to kind of like find the needle in the haystack. So yeah, we are here today. You heard last week from Dr. Sharif Dabdoub about the CHIME and Mother and different other statistical tools that you can use for your microbiome data. And today you are going to be mainly listening to, again, some more of these statistical methods that you can use to simplify your data before kind of like looking at the specific set of filter data and link to your ecosystem output. So this is the outline for my today's presentation. I am going to first start sharing with you some of the resources that I have collected for you. So this is just primers for R and also some links to tutorials for different statistical packages. And then I'm going to jump into how to use network analytics to link your microbial data, your microbial OTUs, your viral populations, your transcripts, your proteins from the proteomes to the ecosystem output. So the health versus disease, the increase in carbon flow, or any other kind of ecosystem feature that you're interested in. And then in the last part, we are going to dive into one of these network analytics. So this is the WGCNA package that I told you about, but we are going to also complement the results that we are going to get from this tool with other methods. So this is the regression methods, such as the partial least square regression, and also the variable extraction methods, so that we can kind of independently evaluate the results that we can get from these networks. So this is going to be a section that we are going to basically go through our code, and understand exactly what are the inputs and the outputs from each chunk of code, and how to kind of the key steps that you want to understand really well and modify based on your data and the complexity of your data and the ecosystem that you are working in with, so that you can get meaningful results. It's not like you have one code that will run for every single data set that you will have, and you will expect that you would get the results without really involvement of the user of these codes. So it's really important to understand what are the knobs, where to actually intervene in the code so that you get meaningful results from your data. So I'm going to start with the resources again. I have here a link from the Center of Microbiome Science webpage. It has here a list of different tools and tutorials that you can consult for learning about R and Python and other things as well. This is a nice list that gets maintained and updated. So I would recommend that this is a good starting place for you to explore. And there are also other resources outside of this kind of link. So you can always go to one of these platforms, Coursera or edX, to learn more about R and Python, which are basically most of the network analytics packages that you're going to see today are available as R or Python modules. So you want to learn more about these kind of like basic tools or basic to learn the commands and like how to handle these software or coding languages. And also you can explore more of the things that are alternative to building networks. So I have here, for example, two links. One of them is for R and the other one is for Python. And these are bootcamp links. You can learn about different things from these tutorials about, for example, using what kind of like the statistical analyses that are appropriate if you have categorical data versus continuous data. And this is in R. And even things that are more complicated, such as using machine learning or building predictive models for your data set. So all of these links are going to be available in the slides when we share them. And they are already also, some of these are available on the page for the webinar from last year. There is also, of course, GOSTAMIA, and this is something that is focused more on the basic concepts rather than the coding or the codes that are, or the statistical packages that are available through R or Python. And this is a really amazing resource because it can walk you through concepts, from really simple concepts to really advanced ones. And it does a really great job of contrasting the different types of analyses that you can do with your data. For example, if we're talking about doing an ordination analysis, so multidimensional scaling, for example, and then you would look at what kind of methods of that is appropriate for your data. So do you want to use non-metric versus metric multidimensional scaling? There's also YouTube. So this is a link for a channel that I really like. It's called StatQuest. The person who is running this channel is a professional statistician, and he does a really great job of simplifying really complex concepts about many of the statistical methods, simple and complex, and they claim that everything is clearly explained basically in their videos, and they are pretty good, pretty well explained in these videos. He explains really well the concept behind the different analyses, and also walks you through the code in Python and in R for how to do ordination, how to do normalization, quantile normalization, and other things as well. And I also have for you here some links to useful statistical packages and the tutorials to learn how to use these packages. And my goal from this is to actually kind of change your thinking about sticking to only one package and using it for everything in your analyses. So Vegan, for example, is one of the very famous packages to do multivariate analyses, multivariate stats for your microbiome data. But if you are just married to only one package, you are missing a lot about all of these other packages that exist out there and can help you and can simplify things for you and can save you a lot of time. So for example, here I'm showing you another package that is called the Microbiome, and this is again a link for the tutorial. It simplifies things for you a lot by basically having these functions that they create in these other packages, and it can do multiple jobs for you at the same time. So if you're using Vegan, for example, to calculate alpha diversity metrics for your samples, you will probably need to call multiple functions, one to calculate Shannon, one to calculate Simpson, one to calculate richness, etc. Versus here, you can only have one line, and it will give you an output that is a really nice table that gives you all of the different alpha diversity metrics in one place. So yes, again, you can contain the same table. You can obtain the same table using Vegan, but you would have to you have to write a lot of lines basically to get the same output. And again, all of these new packages or all of these other packages are made with the out of frustration, basically. So people were trying to do something and they don't find any function or package out there that allow me to do this. So I would have to actually write my own package for that. So there are something like phytosig, there is pathostat. Pathostat allows you to analyze temporal data, so you can look at the variation in the relative abundance of your taxa here, which are on the y-axis across different subjects or across time. And it shows you outputs for you, like really nice figures, something like that, so that you can immediately see the temporal variation or the variation across different samples for your taxa. Chime to R is another package that is more powerful when you look at trees, something like that. So it will allow you also to do some sort of statistics on top of these dendrograms that are outputted. So here you're looking at an example of this color gradient is representing the log2 fold change between different taxa, and allows you, for example, to look at the statistically significant enrichment of different taxa across your tree. There is a lot more, and I'm not going to go over all of these. I just have here again like the links, and based on the type of the data set that you're working with, you kind of want to invest some time and see what's out there, instead of just trying to reinvent the wheel, basically, because most of the questions that you would have are probably solved by someone else in their study, or like they have already written a package for you to do this, to automate this kind of set of analyses. There are specific things here like species easy that I want to highlight. So this is something that is important if you have for the microbiome data analysis, because of the compositional nature of our data. So all of our data are represented as relative abundances. Our OTUs are represented as relative abundances. So this is the compositional nature of the data. And this is a package that allows you to do some of the statistical tests and do ecological networks for spores and composition and the composition of data. There are, again, other things that are focused on trees. There are other things that focused more on building models or doing multivariate analysis, or based on or focused more on doing ordination and multivariate analysis. I have here also two readings that I actually adopted from Dr. Sharif's slides, and they also walk you through a nice set of multivariate analysis that you can use to analyze your data in kind of like any field of microbiology. All right, so for this second section, I'm gonna spend some time again walking you through the reason behind using networks to analyze your data, and kind of like the key differences between the network analysis tools that exist out there. It's not going to be comprehensive. There are a lot of packages that are developed and continuously, and you'll find some similarities between all of these different tools, and you will find also key differences. So again, I'm not here to just like force you on using a specific package, but more of like showing you the key differences between the different approaches, and again, just to contrast the concept behind each of them. So once you walk in and you find like a new package that is published out there, you read the paper and you realize, okay, these are the set of assumptions that they have when they use the network to analyze the data. And this is something that is similar to whatever I have seen before. So when I am going to make an informed decision about I'm going to use tool X versus Y, that's because it suits best the nature of the data that I have. So yeah, again, we are going to look at how to link microbial lineages, microbial functions, viral populations, transcript abundance, protein abundance with the ecosystem output. So if that is carbon export to the deep ocean, or methane flux to the atmosphere, or health versus disease indices. All of that is kind of like different applications that you might want to use network analytics to go about. And the problem, again, as we have been iterating so far, is that how can I relate hundreds, thousands, or even hundreds of thousands of microbes, viruses, transcript proteins to our environmental variables and system output? It is becoming really crucial now because of the scale of the data that people are starting to analyze, global data sets that have millions of proteins or hundreds of thousands of taxa, and it became a key step in every analysis if you are working at that scale. So, okay, so let's go about the concept. So, as I said, you are walking in, you have in your data set a set of samples that you have collected across an ecological gradient. So these are your observations, or M. And then you have measurements that you have from your field. So let's assume that you measure the oxygen concentration on the sample, or the methane concentration or flux, or the water table depth. And these are the different variables that you are kind of trying to interrelate or correlate to each other. So the thing here is that, well, if I want to measure the relationship or understand the relationship between methane concentration and the water table depth, or methane concentration and oxygen concentration, it is easy to do that with like low complex data set like this, because you are basically correlating them to each other or regressing them, regressing one on the other. So let's assume that this is the response variable and this is a predictor here. Or the predicting variable. So you are regressing this against methane concentration. And well, you measure how well you can predict methane flux or concentration based on this from a simple regression model. When you add the microbiome data, when you add your microbial data, viral data, set of proteins and transcripts, you end up having so many different columns. And now you want to figure what is the best predictor, or what is the best variable that you have in your data set that can be linked to your methane concentration or methane flux. So if you are going to really do this individual and pairwise comparison between each different taxon or each different protein cluster in your data set with methane concentration, you will run into a problem that is called large P and small n problem. So you have small number of observations and you have a very huge number of variables. And of course, the problem with that is that you are going to get, by chance, some of these correlations between anything that you have here in all of these columns and your system output. And these are just spurious correlations that are not really meaningful. And that's why people ask for correction for multiple testing. So when you do the statistical comparison over and over and over, you have to correct for multiple testing. And because of that, you end up most probably having a really harsh cutoffs for the statistical correction. And probably it will destroy many of the real significant and biologically meaningful correlations in your data set. So there have been this problem with basically any complex and highly diverse microbiome data set. And because of that, people have been always looking at a solution that is based on, well, let's simplify our data set a bit. So let's look at this high diversity of microbial taxa and try to reduce the complexity in this data set by doing something like dimensionality reduction. And then once we do this simplification, we try to look at which variables within this reduced space that are linked to our system output. So this is the variable extraction step. And here I have an example for that. So this is the standard ordination. This is a principal coordinate analysis. And you know, the basic principle behind this is that we have this multi-dimensional data. So each dimension is basically the set of abundances that we have for each taxon. And you are reducing this dimensionality into only 2 dimensions. So this is the first dimension, the first axis here, and this is the second dimension, okay? And that is something that is capturing around 50% of the variation in your data. But it's good enough for you to overlay your variables on top of this and kind of like try to link the system, the variation that you're seeing in your data with kind of the clusters or the patterns that you are plotting here in the 2-dimensional space. So going back to the example that I started, well, I have microbial data. I just want to see what kind of microbes that are enriched in one cluster versus the other. So in that case, people can just do vector fitting, and they use the abundances of the different taxa here on this 2-dimensional space, and say, well, you know what, we can find out that Staphylococcus are most probably driving the variation here in this 2-dimensional space. And that means that most probably we have enrichment of Staphylococcus in this cluster, which is associated with this diagnosis, the ILD. And that can work, but it can work with severe limitations. So if you notice, we are having here genus level taxonomy. So this is low resolution analysis. And probably this system is also low diversity. So you have only few genera to look at a time. And this is not going to be always the case. So yes, this is something that you can do if you are looking at very low taxonomic or very low resolution. You're looking at high taxonomic ranks, so family, maybe genera, and orders, classes, things like that. But if you are trying to link your species or strains, and if you have thousands of these, it will be challenging and almost impossible to do for like using this as a scaffold, like an ordination analysis as a scaffold. So this is one problem. The other problem that is something that we already discussed, and that is the percentage of the variation explained. So I just told you that, well, the first and second axes are explaining 50% of the variation. So whatever is explained by Cyanobacterium or Staphylococcus is still bound and limited by the percentage variation explained on these two axes on just like this plane. So these are two severe limitations that we have here for using ordination as our method of simplifying the data and doing this vector fitting using microbial data. It can work great with the environmental data. So some people say, well, you know, this cluster is driven by oxygen concentration, or this one is driven by methane flux, and this one is driven by water table depth. So that's great because you already have few measurements from your environmental, you have only few measurements collected for in situ, but for microbial data, things will become pretty complicated. So another way to go about this is to actually pre-cluster your data. So you are basically using one of the many clustering algorithms that exist out there to look at covariation between your microbial data. So you are basically accounting for this autocorrelation between different taxa and building these clusters or modules, and then trying to look at how these modules correlate to your ecosystem output. And finally, look inside these modules and find what are the driving taxa, what are the driving proteins or transcripts of the variation of the whole module. And this is something that we are going to talk about extensively later. So it's more of like a stepwise process, but let's just start with understanding the first step here, which is just the clustering step. So these, again, clustering can be done based on the co-occurrence or the correlation of these taxa or the co-expression of genes. I'm not going to talk about all of these. Again, I told you the goal of this is just to make you understand what kind of decisions that you want to make when you use one clustering method versus the other. So for the purpose of this presentation today, I'm going to do this comparison between WGCNA and MCL, and also WGCNA and SNCC. So I told you that we are trying to build these modules of covariation, and this is how to do it. So let's assume that we have here a bunch of samples, and each line here represents your taxon, your microbial abundance, or viral abundance, your transcript abundance, or protein abundance. So each line of these is representing a transcript, protein, microbe, virus. And again, this is a profile. This is how the abundance profile look like. And this really mess, we want to identify covariation or co-expression. So you deconstruct this mess into these separate profiles. And these separate profiles can be more complicated depending on how many different samples you have collected here on the x-axis, right? And based on these profiles, you try to add or basically bin these lines here to these different profiles. It doesn't have to perfectly fit, but it should be good enough that you were able to segregate your bunch of abundance profiles here into these different cluster sets. So these are your modules, or these are the clusters that you end up having from this step. And again, this is something that applies for pretty much any clustering method that you are using. So yes, so let's do the comparison between WGCNA and MCL to start. Or actually, before I do this, I just want to walk you through the kind of like 3 different steps that I have been going over and over up to this moment, which is basically let's build the clusters, let's understand how to correlate these clusters to environmental output, and then how to look inside these clusters and understand which variables or which microbes or genes or transcripts are important for driving this module, and hence are important to driving your ecosystem output. So WGCNA pipeline or workflow is, again, based on these 3 different steps, right? The first step is the data simplification and dimensionality reduction. And WGCNA stands for weighted gene or genome correlation, co-expression, co-occurrence network analysis. And then it's followed by an independent analysis, which is a partially square regression. And this is something that you do on the modules that are of interest, as I'm going to tell you right now. And this step involves 2 things. You actually do the regression and also do some sort of cross-validation. And this cross-validation is important because it allows you to extract what are the variables that are driving this module variation. And again, I'm going to clarify all of these as we walk through the separate steps. So the first step in WGCNA is building the network. And the way that WGCNA does this is basically looking at the correlation. It does pairwise correlation between each pair of microbes or genes in your data set. And these nodes here, so this is a node, this is a node, are connected with edges. And the strength of this edge is based on the strength of the correlation coefficient between these 2 microbial taxa or genes. So if you have a correlation of 1, you have a very strong connection here or edge here. And if you have weaker connections, so if you have a very low correlation coefficient, then you will have a weaker edge connecting microbe 1 with microbe 4, for example. And after you build this network, this is just an oversimplification of how a network can look like. But if you have thousands of these, you can find a more complex network resulting from this step. What you want to do next is to identify the clusters or modules on your network. It is really easy to identify here, and it becomes, of course, more sophisticated as the scale of the data set increase. So here it's just like, okay, we can basically identify 2 modules, module 1 here, which we're going to call the blue module, and the other module, which they have only 2 taxa here, and we're going to call it the green module. So you identify these modules. Now you want to correlate each module separately to your environmental output or environmental metadata. And this is something that you can easily do by basically doing something like a heat map like this. So you have a bunch of environmental measurements that you have collected from your field site, and you have here your different modules that we are just giving them arbitrary colors. So these are the subnetworks or the modules that you have in your data set. So these are all of the modules on the y-axis, and you have your environmental data on the x-axis. And you look at the correlation between each separate module and the system output that you are interested in. So in this case, we are interested in methane flux, and we found, well, there is a module here, the subnetwork, the orange subnetwork, that is highly correlated, and it have a very significant correlation as indicated here by the p-value. So now, instead of looking at all of these different relationships and the associations that you have in your data set, you just focused on the things that matter, and you just dig into these specific modules and understand better why the taxa or the genes that are existing in these modules correlated with your environmental output. And the direction of this relationship can be the positive direction or the negative direction. So you have here, it can be blue to red or red to blue. So you can have positive correlation or negative correlation with your system output. So the next step is to actually look at these modules. So I just told you that we are only looking at the orange module here from this whole set of modules in our network. So these are the dots from the orange module. So each dot here represents a different OTU, gene or transcript or a protein, okay? And what you have here on the x-axis is what we call the module membership. So how robust or how confident you are that this taxon or protein exists in this module. And this is something that you can kind of like link to or something that is similar to the first dimension in a principal coordinate analysis, a principal component analysis. So things that are here are have the highest membership within your module. And because we are also interested in things that are relevant to our ecosystem, so in the y-axis here, we can correlate the abundances of the taxa within this module to your methane flux or the system output here. So again, now you are only focusing on things on the top right here, top right section. So these are the taxa or the genes on the transcripts that are both relevant to your ecosystem output, and also you are more confident about to be within your module. So these are what we call the hub genes or the hub taxa that are driving the covariation within the whole module. So that all of that was just the first step. So building the network and kind of like having a preliminary idea about what is important within the modules that we have extracted relevant to the ecosystem output. The second step here in the workflow is, again, an independent analysis that is trying to do this regression and cross-validation at the same time. So we use partially square regression or any other kind of like partially square regression that you want to employ. And again, the result from this is something similar to what we have seen from the network. But again, this is an independent analysis that you want to do. You want to have multiple lines of support for kind of like the relationships that you are trying to establish between your taxa and your system output, so that when you go about and do an experiment, you are kind of like walking in with a high probability of success. Or if you are going to, again, invest time in just looking at the kind of metabolisms that are encoded by this specific taxon, so this specific MAGs, then you are kind of like investing time in the right direction. So back to this analysis, again, it's regression, right? So you have here the x-axis and the y-axis. And again, the x-axis here is what they call the latent variables, or again, these are the different OTUs or genes in your subnetwork, or your first ordination kind of like type set of, or first ordination type of laying out your abundances of these taxa. And you are correlating that, again, regressing that against your system output, right? So this is, again, the regression analysis, but then there is an iterative process in building this model, this regression model. And this iterative process involves the cross-validation. There are so many different ways to do this, but the example that I'm showing you here is the leave one out cross-validation. And the idea is basically you construct this regression model iteratively, and each time you just remove one of the OTUs that exist in this module. So you do this every single time, and you calculate the deterioration that is happening in the cross-validated R square between your X and Y, between your X and Y axes right here. So what that allows you is to give relative importance to the different OTUs or the different variables that you have in this relationship. And that allows you to extract, again, the variables of most importance. So this is a variable importance in this kind of projection. So this is the VIP, and this is the correlation, the Pearson correlation, Spearman correlation, whatever correlation that you are trying to establish between your relative abundance of the taxon and your ecosystem output. So this is a correlation to ecosystem output, and this is the VIP value that you have extracted from this cross-validation analysis. And you can also add to that other methods of cross-validation or methods of variable extraction. There is one example here called the selectivity ratio, but this is something that we're not going to talk about at all today. And what you want to do is look for the consensus between the different methods. And the reason behind that is, I told you at the beginning, you can have things that are highly correlated to your ecosystem output, but it's just a spurious correlation. This is something that is a false positive result that you are getting because of the so many statistical comparison that you are trying to do with your data. So if you find a case like this, so you have here a case where you have high correlation, but then a very low VIP score, then this is most probably something that is a spurious correlation. On the other hand, this can also happen with the VIP score. So again, the VIP score is looking at what is driving the module variation. It doesn't really care about the relationship between your variable and the ecosystem output. It doesn't look at all at methane flux in that case. It just looks at within module variation and what is the hub genes or taxa within these modules. So you can have a very high VIP, but that does not necessarily mean that this taxon or this gene is really important to your ecosystem output. So you can have high VIP and low correlation, and this is again a false positive result that you want to exclude. So eventually you want to look at the consensus of the different methods, and this is where you want to focus your effort for more analyses for the metabolism, for example, of that MAG, or if you are going to follow up on your bioinformatic analysis with an experiment in the lab, then you kind of like putting your money on the right place. All right, so that was basically the summary of WGCNA. Again, the last section will be a detailed kind of like walk through the code of WGCNA. But before we do that, again, I want to do the comparison between WGCNA and the other, just like two other examples, to kind of like understand the key differences. So I'm going to do this comparison first between WGCNA and MCL. I already told you about this. We built our network in WGCNA based on the correlation coefficients. We end up having something that looks like this. In MCL, we also build our network. MCL stands for Markov clustering algorithm. And you link all of the nodes in your network with these edges, but these edges are based on the transition probabilities rather than based on the correlation coefficients. The more prevalent use of MCL, if you look in the literature, is for protein clustering. So people, for example, do a blast B between different proteins, and then you would extract the E values of this blast, transform these E values, and use them to build this kind of network. But again, this can be like any kind of like probabilities that you want to have to build these kinds of networks. So that's the first difference. The second difference is the kind of transformations or the kind of data or network modifications that you apply. And this is key difference between WGCNA and other tools. For the transformation step that you're doing on the network, for WGCNA, you are trying to transform these edges by raising them to a power. And you do this iteratively, as we're going to discuss soon. So you raise it to a power of 2, then 3, then 4, etc. And you're going to end up having multiple networks. Which one to select? The one that you are going to select is a network with a scale-free topology. We're going to discuss this with greater depth, but what you want to know right now about a scale-free topology network is that it is this network that if you add more taxa, if you add more nodes, if you add more correlations or more edges, it doesn't change its topology. It has a stable topology. So that is an assumption of WGCNA so that you can actually have anything out of this kind of built network. And the scale-free topology network has something like this. It has like characteristics like this. So if you look at the connectivity in the networks and the frequency, so how many times you see this kind of like connectivity pattern across the network, you should be seeing that very few of the nodes in the network have high connectivity. So very few of the nodes in your network have high connectivity. And you also find that most of the nodes in your network have very low connectivity. So that's kind of like the characteristic thing about a scale-free topology, that this assumption, this kind of distribution in the network. So you're looking at these things that have high connectivity, and they become really important later for module driving, basically. So the taxes that are connected with everything are probably some of the things that are going to emerge as hub taxa or hub genes or global regulator in your kind of like network. So that is for WGCNA. For MCL, it does something similar. It uses what's called the inflation values. And the inflation makes the rich richer, makes the poor poorer. So basically, again, it's transformation on these edges that is going to make the strong edges stronger and the weak edges weaker. The strong edges, for example, if you have an edge of 1, if you raise it to a power of 2, it's going to still stay 1. But if you have something that is 0.2 and you raise that, you're going to end up having lower value after this transformation. So that is kind of like transformation that you make on the MCL. WGCNA then tries to identify the module, as we said before, and this is guided by hierarchical clustering on top of the module identification that we have talked about before, based on the transformation that happened on these edges. So you do this kind of hierarchical clustering on top of that, and the information that you get out of these, the clusters that are going to emerge from this dendrogram, and the modules that you're going to get from the network. And another thing that we're going to talk about in the code, which is basically topology overlap matrix, something that we're going to dive more into in the code. But the information from these different methods allows you to identify the modules. For MCL, it actually tries a more aggressive approach. So it does this kind of inflation and pruning process in an iterative fashion. So it will do a transformation and then cut some of these edges, and then do another inflation, and then it does more pruning, etc., till it reaches a point where you have this sparse network. So a sparse network is basically you have these defined clusters or modules, where if you track each of these nodes, they are linked directly to, they are linked at one point to what we call the sync node. So this is the node that in the center here, and this is the sync node for all of the different nodes within these within this cluster. So because of this aggressive kind of cutting and pruning, we call this process a hard threshold kind of pruning, because again, you are doing the inflation, and then you have a range from 0 to 1 for all of the values that are here in these edges. So if anything, for example, drops below the threshold, that's 0.8, it's going to be removed, completely removed. So that's how you end up having these white spaces, okay? And then you do the inflation again, anything drops below 0.8, you remove it, and so on and so forth. For WGCNA, it actually maintains these kind of like connections, so it will stay in your network, and this is what's called soft thresholding. And that becomes relevant for kind of calculating intramodular connectivity and other things. So this is kind of like information that gets lost on the MCL side, but it gets maintained in the modules in WGCNA, so you can use them for specific things that we're going to talk about later. So that was kind of like what I wanted to convey from this comparison, is that, well, soft threshold versus hard threshold is kind of like has its uses. If you are only interested in the modules on the clusters, and that is your end goal, then MCL might be just enough for you. If you are interested in looking within these modules later and trying to correlate the different numbers within these modules to something or to other modules, then you might actually want to go with the WGCNA in that case. So the other comparison that I want to make is with another tool that's called SNCC, and there are other kind of packages and other algorithms that are also kind of like adopting the same core concepts that I'm going to talk about here for SNCC. And this is again, something really important to understand because WGCNA is not going to work for every single data set that you will have. It is really important to understand how complex your data set is and how the assumptions behind WGCNA are met in your data set. Because otherwise you would, for example, would need to lean towards something else, flash weave, snake, any other kind of things if you have a sparse kind of data set or sparse network. So this is the goal of this kind of like few slides that I'm going to talk about right now, is to understand that you are working always with relative abundance. So this is, again, what I talked about before and told you, this is like the composition and nature of your data. You're always working with relative abundances, not absolute abundances. And these kind of softwares and these kind of like network building protocols are trying to account for this problem, especially in low diversity systems. So trying to look at this kind of sparse correlations and co-occurrence in your network, especially if you are walking in with some sort of compositional data. All right, so let's understand the differences here. So again, WGCNA, you build the network out of correlation coefficients, and that's exactly the same thing that you have for Snake here, which is basically building the network out of these correlation coefficients. The difference between the two methods is that you have control over what you want to use as a method of constructing this correlation and extracting the coefficient from. So you can use Pearson correlation, you can use Spearman correlation, and the default is actually this correlation method, which is the by-weight mid correlation. And it's a hybrid between Pearson and Spearman correlation methods. And the reason behind this is that you are trying to account for the limitations of Pearson and Spearman. So Pearson correlation, you're working with continuous data. But the problem is you have high susceptibility to outliers. So outliers can severely influence the coefficients that you get from this Pearson method. Spearman correlation are less susceptible to these kind of like jumps or like the values that the extreme values that you can get for your correlation because of outliers. But again, that comes on the expense of converting your continuous data into rats. Okay, so the by-weight mid correlation is basically doing this hybrid to account for the drawbacks of like these two different methods. Now, Snake goes with like a totally different approach, and this is a whole kind of line of research that is based on, well, if we are having compositional data, then most probably not any of these methods are not really the optimal method to look at the correlation between your relative abundances of your different taxa. So SPARC here stands for sparse correlations for compositional data. It was initially designed for 16S sequencing data, but it can work for anything else as well. It employs the SPARC correlation for calculating these kind of edges. So the next two slides are going to be focused on this difference between why SPARC is actually better than using Pearson in some cases for calculating the correlations between different taxa, relative abundances of different taxa. So this is the this is kind of like the study that is trying to lay out the reasoning behind developing SPARC. So you are having here compositional data, your microbial 16S data from different ecosystems, and you want to compare the Pearson method versus a SPARC method, okay? And the way that they did this is basically they have simulated data. So they walked in knowing what relationships are real and what relationships are false. So they walked in with simulation data. This is not natural data, okay? And they try to compare these two different methods by basically calculating the error, how many times you are basically getting a correlation incorrectly. You're getting a false positive correlation, false positive hit. You're getting correlations that are spurious, that are not meaningful. And this is Pearson here on the left, and this is a SPARC on the right. So basically, as the amount of error increase, you get these darker colors, the red and the black ones. And this is a very clear like visualization of the difference, right? So you find that pretty much most of the box here on the left for Pearson is in the tinge of red to black. Yeah. And that means you have a lot of error that can be accumulated in your kind of analyses because of just using an incorrect method of doing this correlation. Versus Spark here, you find that most of the box here is actually on the light yellow side, and might be like also some of like the darker yellow color. Yeah. So that's great, but also you want to pay attention to something. So you have here another two axes. Yeah. The first one here is about the density in the network. So how many connections that you have in the network. And on the y-axis here, you have something that is called N effective, which is the number of effective taxa. So that is a measure of the diversity. It's an index of the diversity of the system. And this is something that I'm going to explain more on the next slide. So it's really important to understand this. If you have a data set that has very low diversity, like the vagina or the gut, then you are running the risk of having so many different wrong inferences in your analysis if you are using Pearson correlation. In that case, you have to stick to Sparsy or Spark. But if you are working with a system that has high diversity, most probably you'll be fine using either method. Okay, so this is a clear example of how diversity impact the inferences that you make. This is again from the same paper. They have here their basic network. So this is the simulated data that they have. This is the correct answer that you should get. And this is the answer that you're getting from Pearson. And this is the answer that you're getting from Spark. And here on this axis, you are looking at the more complex data sets as you are going down the axis here. So this represents, again, the N effective or the diversity of the system. So there is a formula for that. There is a formula to how to switch between Shannon H, for example, the more common metric that you use for estimating diversity, versus this number that they have, the number of effective taxa. And the formula is this: you have this e to the power of your Shannon's index. So if you have a Shannon index of 2, this is the N effective that you have, and its location is right here on the y-axis. So if you try to run Pearson correlation, you look at this column and you find all of these extra linkages in your network that are spurious, that are not real, versus Spark that actually accounts for all of these and gets you something that is really close to the correct answer or the original network that is built. You increase the diversity in the system. So this is Shannon's H of 3. So that gets you to around 20. You still have some spurious connections in the networks from Pearson correlation versus Spark. But then you increase the diversity, Shannon's H of 4, you get to 55. So that is basically out of the scale that we have on the slide. And now you have correct network predicted by both the Pearson and the Spark methods. So again, all of that to say, you have to understand your data. When you find that somebody says, oh, actually, you should be using another tool, you should be using FlashWeave instead of WGCNA, and you already did the analysis and you actually don't have any problem because your system is diverse, that will save you a lot of time to actually just like jump between different tools based on personal experience. So you're making actually informed decision about what you're doing and understanding that is the really correct tool for your data set. One more thing is you can also modify the code. So whatever tool that you're using, you can transfer the good bits from other tools to your workflow. So people have been also doing modifications on the WGCNA workflow by transferring some lines that will calculate Spark correlations and replacing basically the coefficients in the WGCNA workflow with the coefficients that are calculated using the Spark workflow. So it's possible, you know, like you want to be kind of like in control of what you're doing. All right, so now let's go back to the main workflow of WGCNA versus SNCC. So we calculated our network using the two different methods. We did the transformation for WGCNA. Again, the goal is to get the scale-free topology network. So most of the nodes have low connectivity, and very few of the nodes have high connectivity. SNCC doesn't really assume any scale-free topology. And this is something, again, I have here for kind of like our understanding is that this is an assumption, right? From the people who develop this kind of workflow. Does it really apply to every single system and every single biological question? We don't know that. So this kind of assumption is not necessarily fit for everything. And that's the kind of like the understanding that they walked in with like developing this package, the SNCC package. So they do not assume any scale-free topology. So that's like a good thing, but it's also a bad thing because again, you are missing on the kind of like intramodular connectivity relationship calculation that you can get from WGCNA. So you ended up having, okay, really nice clusters from these networks, but you might not necessarily be able to look inside these modules and extract specific members based on their relative importance. Okay, next we said that for WGCNA, we look at hierarchical, we apply hierarchical clustering and we calculate the topology overlap matrix so that we can identify the modules. In SNCC, it also does hierarchical clustering. It's a little bit different because it does like single linkage versus average linkage. And again, it's hard thresholds. It's mainly focused on getting the modules rather than maintaining the relative relationships with of the different taxa within the same modules versus the soft threshold that we talked about for WGCNA. Okay, so I have some notes that I want to close out with. I have been talking about this intramodular relationships and how important these are. So in the next few slides, I'm going to be talking about why this actually matters, why these intramodular connectivity matter at all. There are several reasons. The first one is that, as I told you before, that this is something important to identify what are the hub genes or the hub taxa within these modules. It's not, again, about just creating modules. It's more important about this connectivity because you get these eigenvectors or eigengenes, as they call in this statistical package. And these eigengenes allows you to do a lot. It allows you to link your module with other modules in the same network, or your module with other modules from totally different networks, or you can use this module and link it to your environmental data. So you have something that you can quantitatively link to so many other things. And that's kind of like the importance from their perspective of these intramodular connectivity preservation and the modules that were generated for the WGCNA. So what are these eigengenes or the eigenvectors? It is a measure of the expression strength or the relative abundance importance. And again, this is a thing that we have been saying before about like the first axis when you do this kind of regression and say, think about it as a first axis of a principal coordinate analysis. So this is the relative importance of the taxa or the module membership, how important this taxon in this module. And it is basically calculated as the weighted average of the expression of or the relative abundance of all of the genes or the genomes within this module. And they claim that this is something that is very robust and regardless of how aggressive or how lenient you were in identifying modules. What do I mean by that? So as we walk through the code, you will find that you actually have the power or you have control over how fragmented the clusters are or how coarse the clusters are, how big the modules are or how small the modules are. So what they are saying is that this is also a safeguard. This is a layer that allows you to amend any mistakes that you have done during building the network. If you were really aggressive and you ended up having so many small modules, you can link these modules back based on these eigenvectors, and you have a bigger module. So this is something, again, I'm going to show you which step exactly in the code you have control over. So again, they are saying that, well, you have these hub genes. It will allow you to connect your different modules to each other and other things. Where to find these hub genes or hub taxa? If you are trying to represent your data set as a tree here, and this dendrogram here, and you build this kind of like you did hierarchical cluster and look at this, they are the taxa or the genes that are at the tips of these branches right here. So these are the things that are driving these modules, okay, the respective modules. And in another representation, if you are looking at an ordination analysis, so multidimensional scaling, what we're looking at here, and you will find different clusters that they are the taxa or the genes or the proteins that are at the extreme ends, at the fingertips here of these clusters. Okay, we already talked about this. So we already said that the eigengenes of the eigenvectors are important because it allows you to relate modules to each other in the same network or across different networks. So this becomes important if you have multiple data sets. You have metagenomes, metatranscriptomes, and metaproteomes. And you build a network out of each of these. You can actually link the modules to each other using the eigengenes. How the module of covariation at the gene level is related to the covariation at the transcript level and the protein level. And that is a whole set of interesting things that can come out of this. So are we seeing that most of the system output is driven by the mere existence of the function, or how we'll express the function at the transcript or protein level? So there are so many interesting biology that you can explore by just doing this kind of like comparison from these values. So these are important things to have in your hands. Again, you can correlate that to your environmental variables, physiological data, clinical traits, etc. And one important thing as well, again, we talked about the scale of the data nowadays. So you can actually have hundreds of thousands of genes or proteins in your data set, and trying to build a single tree. If you want to do hierarchical clustering and build a single dendrogram out of all of these, it will be very computationally demanding. So one of the things that the solutions that they provided in this package is to break down your data set into these blocks. So there is a function that we're going to look at. It's called the blockwise modules. You break your data set into blocks, and then you run each block in parallel. So you use different cores in your machine to run the independent blocks and build a tree for each one of these. And then you can connect these modules using these eigengenes or eigenvectors at the end with each other and get the final kind of module. So yes, and finally, this inter-module relationship allows you to identify what are the hub genes, what are the hub taxa, and highlight their module membership. This is an example of how to connect two modules. So these two modules here, the blue and gray, were actually pretty decently correlated. So that tells you that, well, actually you had severe, at one step of your analysis, you were harsh with your trimming, and you ended up having a single cluster that's split into two. So using these eigengenes can allow you to merge them, so add them to each other. So yes, look at the major assumptions of your tool that you are using, and the nature of your data set, and base your decisions on that, the fork flow, and also key steps that you are controlling in across the pipeline, so that kind of like you don't walk in with like incorrect assumptions and end up having something that is just totally spurious connection that is not really biologically grounded. one more thing here that I want to share with you, and this is really important as well. There is assumptions about the modules that are built from these networks. And the example that I have here is WGCNA. Should all of the members of the same module co-vary in the same direction? So should be all correlated, positively correlated? Should all of them be negatively correlated? Or is it okay to be unsigned, which is actually the default thing of WGCNA? So make sure if you're using WGCNA to understand what kind of network that you want, signed or unsigned. The default is unsigned. So the developers actually use the signed network more than the unsigned network. Unsigned means you build a module and things can be positively correlated or negatively correlated. That again goes back to your biological question. Am I expecting that all of the things will change together in the same direction? So do I have symbiotic relationships? And that's what I'm interested in. So am I looking at the organism and all of the symbionts of it? So in that case, maybe you want to focus on the signed network. Another case, if you are looking at regulatory networks, so you're looking at genes and you know that there are global regulators that, since they are global regulators, they are most probably will be the hub genes on your modules, and they can increase the expression of other things or downregulate the expression or downregulate other genes or decrease the expression of other things. So in this case, you want to understand that, well, unsigned network will be most probably the better thing to go with, because you want to have your module built off your global regulator and everything that it interacts with, it controls. So make sure that, again, you kind of like not just run the code, but to also understand what you want to have as an output from running this code. All right, so this is the last part of my presentation today. Is we're going to go through the 3 steps that we have discussed: building the network using WGCNA, using partial least square regression, and extracting the variables. And this is a code that I have here. It's already shared online, and it is modified from the code that they used in Guidi et al. 2016 to link microbial taxa, thousands of microbial taxa, and viruses, and also eukaryotic taxa to carbon export. So they wanted to understand which taxa are most probably driving carbon export to the deep ocean. And that was a really hard question given the nature of the data. It was spatial, temporally confounded, and it has so many things going on with that. And they wanted to understand how within this whole complexity of data get meaningful results and linkages, and see and like test that against the literature and seeing whether we have any biological or ecological support for these things. So that's again the use or the utility of having multiple lines of kind of like investigations in your code. All right, so we are going to start with this chunk right here, which is basically straightforward installing the packages if you are running this code for the first time on your machine, or if you have all of these packages that are pre-required to run the code in its entirety, you will be just like loading these packages to your system, to your kind of like to your R console, to your R session. One more thing, that this is something that is not actually told to do by the like it's not necessarily something that you will have in if you run WGCNA, and it will severely limit kind of like the machine capabilities if you are just going in through the code without enabling multi-threading. It will only use 1 core of your machine. So things, if you have 8 cores, so most probably things will run 8 times slower than if you enable the WGCNA threading. So it will allow you to use all of the cores in your machine. So yeah, that's pretty straightforward. Then this chunk here is about importing the data and just like setting your work directory. The data that is shared with you here is not my data. This is data that I have found online, kindly provided by the Bowman Lab. They have this 16S data that they generated from polar samples. And they have also some metadata. They have some environmental measurements in situ. And this is a perfect data set actually to look at because of like, you can do this kind of like build the modules and compare them to the metadata that they measured and all of that and do regression and variable extraction as we're going to see today. This is kind of like how their data looked like. It's, again, this kind of bunch of lines is about reading the table that they had on their website. Again, I'm just showing the first 5 columns and rows in this line here, 33. So this is how it looks like. You have the samples, 1, 2, 3, 4, 5 samples, and you have your OTUs here as rows, OTU 1, 2, etc. And you have here the counts for the 16S data. The next step is just to transpose the table. So make your OTUs as columns and make your samples as rows. That's it, because this is how the WGCNA imports the data. It assumes that all of your taxa are the columns. Again, these are the variables, the P, and these observations that are the N. So it should accept it in that in that form, in that format. The next step is to transform this table. So we have here our taxa and we have the samples. Because WGCNA assumes that it was originally developed for expression data. So it assumes that you always have some sort of expression, background expression. So even if a gene that is completely turned off, there is some sort of like, you find one transcript or something. So that was the assumption of WGCNA, and that's why you want to do this kind of transformation if you are working with relative abundance data, because actually in your data set you will find the opposite. You'll find most of the taxa have zeros here. Okay, so it's not existing in many of your samples. And because of that, you want to do this transformation so that you can give lower weight to the absence of taxa in these different samples. The method of transformation here is the Hillinger transformation. This is a function to do it, the constant, and the method is Hillinger. So Hillinger transformation is doing a square root transformation of these relative abundances. And this is how it looks like. So now, instead of having very high values like 1,700 something, it's now 0.4. And it can be basically closer to the zero values that we have right here. And hence it gives, again, like lower weight to the variables with very low counts or with so many different zeros. Okay, line 45 here is something that you don't have to do. This is really important to know. In the code that is shared with you, there is already a step that tests for the samples and the taxa that are very low represented. So if you have zeros across for a specific sample, or have zeros across for a specific OTU across all of the samples, this is going to cause a problem for the code when it tries to build a network. There is already a proof line that is trying to correct for this. It excludes all of these different cases. So that's why I'm saying you don't have to do the step 45. This is something, again, from the Bowman Lab, the code that they had. And they were basically, because they have looked at their data, they were seeing that, well, if we have anything that has a relative abundance below 0.05 after this transformation, they are going to remove that completely. So they are kind of just like cleaning their data, removing anything that is rare in their data set and just focusing on the abundant taxa. So this is the step where I told you where we check for if there are too many missing data. This is a function from the WGCNA package. It's called good samples genes, and it takes your transformed table, the Hillinger transformed table, and it looks through it and it tells you, well, should you proceed or not? This is the line that you write to see if your data set is ready for building the network or not. If it's not ready and if there are so many missing data, it will tell you it's false. If everything is okay, it will say true, and that's how you proceed. So if it gives you a false output, that means you need to remove all of these samples. And again, there is a bunch of lines here in the code that allows you to remove all of the samples and the genes that are basically having 0 counts. It has here this exclamation mark, which means not larger than 0. So if it has 0 across, for samples or genes, they are going to be removed. Okay. So this is just like, again, cleaning your table, and then you test again. And if it gives you true, then most probably you are ready to go. You remove the offending samples, you remove the offending genes, and you still have enough data to proceed with building the network. One more thing to do before building the network as a quality control is to also look at your samples for outliers. And this is basically by doing a simple hierarchical clustering. This is line 71 here. You do this hierarchical clustering on the distances within your data set that is Langer transformed. And you look at the output from that. So this looks okay. Apparently, we have here 1 cluster, 2 clusters, 3 clusters of samples. 3 clusters of samples, not OTUs. And that tells you, well, you might, if this is an ordination, probably you'll find 3 different clusters in your 2D space. So that's okay. It tells you there is a pattern in your data set. That's fine. The problem will emerge if you have a strange topology of this kind of like dendrogram. And by that, I mean you find, for example, an outlier. So there is a sample that is on its own here. This sample might cause you trouble when you try to build a network. So when you do this and look at the data, it will allow you to, okay, I'm going to exclude this sample. And of course, you provide reasoning for why this, and you look more in the samples, like about this collection, about the sequencing, about a lot of other things, whether technical or biological reasons for why this sample is behaving differently. Maybe it's actually like a forced cluster, but you don't have enough representation of this forced ecological feature of this kind of set of samples. So it gives you an idea about like how to proceed with your data set before investing time in trying and forcing everything in a network that is actually not going to be representative of anything. All right, so now after you did all of this cleaning, you want to build your scale-free topology network. And again, we talked about this scale-free topology network, and we said that we build it by doing this iterative transformation on the edges, do power transformation after one after the other, and then you get all of these networks, and then you pick one of them that's meeting the assumption of scale-free topology. So this is how you do it. This is how to build a network. This is how to plot the different networks, the different indices of the different networks, and decide which network you're going to pick that is fitting the scale-free topology. So the first line here, the 84, line 84, is you provided with a vector of power transformations. So power of 1, 2, 3, up to 10, and then you jump by 2, 12, 14, 16, etc., up to 30. So this is the jumps that you are making. And this is the function that you have, which is pick soft threshold. So this is all of the powers that you have specified here, and you apply it on your transformed data to build the different networks. And here I'm just assuming that we're gonna do signed network rather than unsigned network. Okay, this has to match then all of the steps that follow. So if you choose to work with signed network, all of the steps later has to be signed. If it's unsigned, all of the other steps have to be unsigned. If they are 2 different things, then you are most probably looking at 2 different networks. So you pick the network based on this step and you apply it for later steps. If you are choosing signed here and unsigned later or like elsewhere, then you are actually comparing apples to oranges, okay? So that's what's happening here in this step. And then you are plotting two things. You are plotting the R-square value for how the network is fitting your scale-free topology assumption. And the other things is the connectivity within the networks that are generated. So these are two plots that I'm going to show you in a second here. This is the scale independence and the mean connectivity. Again, y-axis here, we're talking about the scale-free topology assumption fit. So this is the R-square value. And we have here the mean connectivity. And this is the vector that you have specified from 1 to 30, 1, 2, 3, 4, up to 10, and then you jump by 2, 12, 14, 16, etc. And these, again, are the same values that you have specified. So what you're looking at here is that as you increase the power, in this case, you are actually meeting the assumption of scale-free topology better as you increase the power. This is not going to be always the case. It can be all over the place. Sometimes it will be, for example, 9, 10, 12, and then it will deteriorate. This is just like an exemplary result that you can get from WGCNA, but it's not going to be as beautiful like this every single time. And that is telling us, well, if you pick something like 8 and above, most probably you have met the assumption because the R-square value for this fit is 0.8 and above. So which value to pick? Am I going to pick 24 because this is the maximum? Yes, this is something, well, I mean, yes, it is the highest fit with the scale-free topology assumption, but it's not the best value to use for your network. And the reason behind this is that let's look at 24 and the other plot. 24 is here at the close to the end of this tail. And this is the connectivity. So as you increase the power transformation, you fragment your network more and more and more. So by that, I mean, you might end up having a network of just single nodes at one point, just like individual nodes. And that's not a network, right? You just basically fragmented your network into their individual nodes. So you want to pick a value that is high enough to meet the assumption of scale-free topology, but at the same time, it's low enough that you still have some sort of connectivity in your network so that you can identify modules small or large. So in that case, the rule of thumb is that you look at where the kind of like this trend slows down. So you look at the shoulder here. So you can pick 10 or 12. In that case, we will pick 10 because it will have around 8 connections. This is as a mean connectivity in your network. So 8 connections per node. So again, I told you we picked 10. So now this is the value that we picked. And this is the value that we're going to use for building the actual network, building the topology overlap network and finding the modules within it. And this is a very important step. You want to understand how you control the different parameters in this step so that you can get your modules that you are interested in and use them for the different correlations down the line. So this is the function to build the network. It's called, again, the clockwise module. And I told you it's called clockwise because you can basically build this network into blocks and then join them together. You give it your transform table of Wrangle transform table. You give it the power that you have selected, which is the 10 here. So this is the specific scale-free topology network that you have picked. And you specify the type of correlation, Pearson, Spearman, or mid-Biot correlation. You specify the maximum block size. So if you have a humongous data set that is hundreds of thousands, maybe you want to say, well, let's divide it into chunks of 10,000 taxa in each block. Or if your data set is small, then you basically set this number to be more than the number of taxa in your network, and then you end up having everything in a single network. You specify the network type, signed or unsigned. You specify the topology overlap matrix. That's what I told you about, whether it's signed or unsigned, it has to match. And topology overlap matrix, as I told you before, is another layer of the decision making that's happening through the algorithm on identifying these modules. So it doesn't just look at the direct connections, but also the connections beyond. So the intermediate connections. Consider the intermediate connections as well with nodes, other nodes inside the module. And when it does this, it kind of like also tries to reduce the spurious kind of connections in these modules. You can also specify the minimum size for your modules from this network. So are you going to accept a module of 5 members or 5 OTUs? Maybe that's fine. But then if you have very few members in your module and you're trying to do regression with 5 data points, then most probably the statistical, the kind of like the power of this kind of like test that you are going to do later is going to be very weak. So you want to have a value here that is decent enough so that you can kind of like for model building and for correlation with environmental variables and correlation with other modules is high enough that you can actually have a robust statistical characteristics of this kind of model that you're building. The line here, which 182, this is about the reassign threshold. I told you that this process happens iteratively, and there is reassignment of OTUs to different modules in each iteration. So when you give it a 0, you are basically given the freedom for the algorithm to reassign the OTUs to the different modules. So if OTU 1 was assigned to module 2 in the first iteration, and then in the second iteration, it was best fit or best assigned to another module, module 5, then you give it the freedom to actually move this OTU from one module to the other. The line here, 129, is specific to something called the dynamic tree building. I told you that there is hierarchical clustering, there is a topology overlap matrix, and there is the network that you built, the scale-free topology network that you already walked in. Yeah. And if you are, this hierarchical clustering can be standard or can be this hierarchical clustering that you applied, you want to basically identify what are the clusters resulting from this dendrogram. So if this line is basically not commented out, it will allow your tree to be dynamically trimmed, okay? And you have values here from 1 to 4, and based on that, you kind of like assign the aggressiveness of trimming that you have in this dynamic tree cutting methods. If you want to have more control, you can actually apply a specific threshold for where to cut or merge the tips within your dendrogram. So this is a merge cut height. It's a low value here, which is basically saying anything that is below 0.1 is going to be merged together in the same module. Line 131 here is talking about what kind of like labels you want to give to your modules. So by default, it will give you, if you assign this to false, it will give you colors for the modules, green module, blue module, brown module, and instead of giving them module number 1, 2, 3, 4, etc. And for this step here, this is something that you basically try to give relative power for the dendrogram from the hierarchical clustering versus the partitioning around the medoids, and that is from the topology overlap matrix. So whether this should respect that or not, so you said it like false. So you give the freedom again for the partitioning around medoids over the hierarchical clustering. Because again, I told you hierarchical clustering can be tricky with really massive data sets. So this is like in that case, we want to set it to false. You want to save the topology overlap matrix right here. And that is because this step takes the most time of the pipeline. And you don't want to run this every single time. So if you actually save the topology overlap matrix to your system, you will be able to import it at any given point and just like do your analyses rather than having to wait for hours, sometimes days, if the data set is really humongous, to regenerate this topology overlap matrix. This line right here is something that you want to activate if your data set has a lot of missing data. It tries to correct the network for so many missing data after the initial cleaning that already happened in the earlier steps. Verbose is just like it outputs messages to your console screen as you are running the system. So it tells you iteration 1 is done, iteration 2 is running, etc. And you also just like give a prefix basically to the modules that are generated from this network building and module generation step. All right, so you finish this, you build the network, you got your modules. The next thing is to start to look at, okay, which modules matter. This line here is something that just like I prefer to do is just like, reassign the colors for the module so that like I remove any stigma related to the gray module. This gray module is something that is where all of the taxa that are not really assigned to one of the other modules will fall into. So this is a collective thing. And sometimes they are actually useful. So sometimes you find that this is your hub basically that's driving the whole network. So this is again just the line that's assigning the colors and saving them in this variable here. And you want to do the correlations. So this is the time that you want to load your metadata. And this is again the metadata that they collected in the Bowman lab. You import that. And you want to calculate the eigengenes or the eigenvectors for your modules. And this is the function, line 147 here is the line that you use to do this. So this is the function that you use on your Wenger data and basically the different modules that you have here, and you extract the eigengenes from that. These two lines here is just reordering these eigenvectors so that like they are put next to each other based on their similarity on the profile of the eigengenes. So now we have this kind of vector pops here. It's gonna pop up like in the coming lines. So this is the vector, the matrix of the eigengenes. Okay. So we got our metadata. We got our eigengenes that we are going to use to correlate our modules to the metadata. Yeah. And this is basically how to do it. So this is, again, correlation function, standard thing. And you are correlating the metadata matrix with your POPs, which has this is a matrix that has your eigengenes, and you're using a standard Pearson correlation method. You also try to extract the p values. So this is a function, correlation p value, and you are calculating that on your variable right here. That is basically the correlation, that is the output of the correlation between your eigengenes and your metadata. And of course, you give it the number of samples so that it can calculate the degrees of freedom. So you did all of that, and then you plot. You just plot. So all of this chunk here from 159 to 173 is just plotting all of that, and it saves that to a PDF because you don't really know how big the metrics will be, and sometimes it will not fit into your viewer in RStudio, for example. So you export all of that to PDF, and this is something that you might get. So you already saw this before. So we have here on the y-axis the modules, the subnetwork, blue, turquoise, brown, and yellow. And you have here the different environmental data, the metadata that they have in their sheet. And you are trying to scan this heat map for the strong correlations, either in the negative direction or the positive direction, and also has a significant correlation. So now you know, well, what are the relationships that you're going to ignore, and what are the relationships that you are going to focus your effort on, or at least start with and then go later to other things. So what we want to do from this is we're going to extract two things. Okay, so we're going to assign two new variables here. Brown, this is the module that we are going to investigate further. And this is the environmental parameter that we're going to investigate further, the upwelling here. So now you want to extract the values of your parameter. Again, the parameter is the upwelling. you want to extract the values for this parameter out of your metadata sheet. And this is now just a vector. This is just, sorry, it's in a data frame. This is just the values that of your ecosystem output that you're interested in, okay? And after you get that, you want to clean the data again. So we cleaned the data already for your relative abundances, but we never clean the data for the environmental variable, because basically you can still also go to if it's satellite data and it's a cloudy day. So maybe on the date of collection, you didn't get this environmental measurement achieved. So you're now missing some data. And you want to also clean this and make sure that the comparison now that you are going to make between the metadata and the module members is not influenced by is actually correct. So you have like matching basically samples and across the two tables. So for example, line here, I'm not going to go through all of this, but this line here, 186, is removing all of the missing measurements, DNA on MET for like these parameters, okay? And you basically check if the dimensions are the same across everything. So you have here the clean table for the Hellinger transformed data. You have here the clean table for your metadata, which is weight right here. And this is MB clean is basically something that is coming from your environmental variables. Sorry, you're coming from the eigengene matrix that we had from before. So everything is here is matching. Now we can go forward with our further kind of like relationship and investigation. All right, so I told you before that we are the first thing to get out of these modules is kind of like correlate the module membership with the metadata, the correlation with metadata. So this first chunk here is basically trying to do this correlation between your Hellinger transformed data and your eigengenes. So this is the x-axis. And the other chunk here is trying to do this correlation between the clean data for Hellinger, because now we removed all of the samples that are missing environmental data and your environmental data, again, using Pearson nest. You get these two, and then you do plot them against each other. So all of that is basically just like a bunch of lines for plotting a verbose scatter plot, which is basically this plot right here. This plot right here has here, again, the significance of this specific OTU, all of these dots, different dots. These are the specific dots of specific OTUs that are related to the upwelling. And this is the membership in the Brown module that we are interested in. Again, the things that you want to look at is in the upper right corner here, things that are strongly belonging to this module and also has high significance or high correlation to the upwelling. This is a pretty good thing. It's almost linear relationship, and you find this is a decent correlation and a lower p-value. But again, this is just the WGCNA. We said that we want to do some independent analyses to kind of like look at this direct relationship between your taxa within this module and your ecosystem output. So this is when we do the partial least square regression. And we walk in with some assumptions. So we are going to say, for example, I'm not going to look at anything that has an R-square below 0.3. Okay, I don't care about anything that is below the threshold. This is the baseline. I just want to look at anything that is above that. Of course, you want to be like at the output, you want to be looking at really high things, 0.7 or above, but just like as a screening step here so that you don't have to run a whole lot of data through your regression, partially square regression analysis. So this is the step where you actually do the partially square regression. This is a function. You regress two things against each other. This is the subnetwork here, which is again your Hellinger transformed data, but not all of the OTUs, just the OTUs that belong to your module of interest. And you regress that against your environmental variable of interest, the upwelling. And you specify that you want to do this cross-validation, the leave one out cross-validation, and use the orthogonal score, partially score regression. Orthogonal scores should be something that you already know now, because in the ordination, each axis is orthogonal to or each dimension is orthogonal to the next dimension. So this is the first dimension and second dimension, and then the third dimension, fourth dimension, etc. So now that we have done this, we can look through the dimensions. So in principal coordinate analysis or other set of these kind of like orthogonal based methods, you will find these different components or different dimensions. So you want to screen through the different dimensions and look for the R square that is high enough. But again, you're not going to go all the way up to the all across all of the dimension, because that defeats the purpose. You stop here at one point where you have the maximum R square with the smallest number of dimensions. And this is something that does automatically. You don't have to specify this. So this is the utility of this for loop right here. It will go through all of these components and it will extract the highest R square. And it will tell you basically the maximum R square found was 0.58, and that is corresponding to the seventh dimension or component number 7. During this step, you also try to get your variable importance and projection values. So this is something that you basically call the code for. This is the line 251. You get the source code for it from this link, and it should be able to calculate that for you in later steps. These bunch of lines is just like telling you whether to stop, because actually you didn't meet any of the assumptions, like the R square value was below 0.3, so just stop there, don't go forward. Or if we actually found a decent R square value at the lower dimension, relatively lower dimension, that means we should go forward with calculating the VIB. And this chunk is really big. I'm not gonna go through all of it. What I want to say is that there is a specific step that you calculate the VIP score through if you're running this through this part right now. And it also, you can specify how many of the taxa you want to look at. So in the case that we looked at, we had so many dots in our kind of correlation analysis, and when we looked at the module membership versus the relationship to module to upwelling, there were so many dots. We don't want to look at all of these. We only want to look at the things that were on the upper right and look at like the top 100 or something. So instead of just like again diving through a lot of data, and you can also export the VIP scores and the Pearson correlation, pair each of these OTUs to your system so that you can kind of look at them later. So this is something that, this is how it will look like. So this is the OTUs, and this is your VIP scores. This is the OTUs, and this is the Pearson correlation. And also you can have this file here, which tells you basically the ranking of these different OTUs. So the OTU with the highest rank was here, OTU number 5, because it has the highest VIP. So maybe this is the hub of our module. Okay, final 2 slides. What are we going to do with all of this data? So there are 2 important comparisons to make. The first comparison is we built a model out of partially square regression model. We built this model. We want to compare our model to the actual measurements that we have from the field. So if we use our relative abundance data or gene expression data to predict the ecosystem output, is it really meeting the measurements that we have made in the field? So this is one thing. The second thing is comparing VIP score to normal correlation. And this is again important because I will remind you again, normal correlation can be spurious, VIP score can be false positive. So we want to look at both of them at the same time. So the first one here, again, the comparison between measured versus predicted. It's pretty straightforward. This set of lines is just a way to plot the equation, the regression equation on the figure, so on the plot. So there's nothing really important here or specific here. And this is just like the 3 lines that you use to do this kind of regression. And this is the bunch of lines that you use to plot and beautify the figure. And this is the figure that you want to look at. And again, it's not going to be always as beautiful as this. And I'm saying that this is beautiful because you look here at the blue line, which is the regression line, and compared to the black line, which is the one-to-one line, and they are very close to each other. They are tracking each other. So that is telling you that your model is pretty quantitative as well. So whatever things that you are going to predict using your relative abundance. So again, the beginning goal was to link your microbial data to the ecosystem output. If you're saying that this microbial data is really important to ecosystem output, and you do this regression model and cross-validation, all of that, and you want to predict the ecosystem output just based on the relative abundance of a specific taxon, and you get a result like this, well, it gives you more trust and also allows you to do kind of like scaling if you want to build prediction model under changing climate, under changing disturbed environment, or any kind of like scientific question that you're after. So this is an important result to get out of this pipeline. Kind of like also the characteristics of this relationship is high, R-square value, and the p-value. So that's one thing. The other thing is to compare the VIP scores to the Pearson correlation. And again, this bunch of lines is just like collecting the data and putting them into a single table. So nothing really specific here. And you end up having a table like this, which is basically the OTUs ranked based on their VIP score and their normal correlation. And this is a clear example of where I said you can have really high VIP score, but very low correlation. And for OTU 11 and 26, it is more acceptable here because we're looking at high VIP score and relatively high Pearson correlation. There is also another case here for OTU 1, OTU 2, like pretty much there is no correlation whatsoever, but it has a high VIP score. So this is kind of like the table that you want to have so that you start to look at the specific taxa, either their metabolic blueprint, or if you want to do an expression analysis on the bench or anything, you want to start kind of like prioritizing the taxa that you're going to look at. So yes, that's what I have for today. And next week, you're going to hear from Dylan Cronin about the building mags and the quality control steps that are involved from getting the reads up to high quality mags. And then you are going to have a month of break, and then we're going to continue with the rest of sessions for this microbiome webinar series. I am going to stop sharing now, and we are going to start our Q&A session. Thank you so much. So I have here two questions in the Q&A. I have here, will the slides and the script to be made available? Yes, the script is already available. We are going just to move it from last year kind of resources to this year so that you can easily find them. And the slides as well, I can make them available. Of course, the recording is going to be available as well. I can provide especially the set of slides that have the links for the different resources, like for the statistical packages and their tutorials. There is also another question, is line 153 where you could substitute code for Spark? Yeah. I think this is where you can do that, yes. So this is basically the line where you specify whether you want a Pearson Spearman or the mid by weight correlation. It's not really in code, and it's not really embedded in this code, but 53. So Renee, I would have also to see exactly where people have done this, because I think 153 is something that is coming after building the network. So I would assume that this is a step that has to happen before building the network itself. But I can go back and look through the different codes that I have and things that I have seen and see how they did something like that. So I have also a question, the data are compositional, why Pearson, not Spearman, is used. Well, to just like, in that case, we were trying to look at the data in not like in rank-based fashion. I would assume that you want to preserve this kind of like continuous nature of the data for building models. That's my assumption. And I think it would be even better to use the mid by weight correlation or using something like that Spark. But I think for the data, it was complex enough that Pearson was working fine. No, thank you so much, Diana.
Ahmed, I actually have a question for you from the slides.
Yes, of course, Sri.
So you had mentioned in your WGCNA versus SNCC slide, you had mentioned that one of the reasons to potentially choose SNCC was for data that wasn't very highly diverse. But then you also mentioned, so in other words, would that go away if you chose the appropriate correlation metric? So if you switch to the sparse CC or something else?
I'm assuming yes. I'm assuming yes.
Okay. The other question to follow up from one of the Q&A questions is why you chose the Hellinger transform as opposed to like the centered log ratio or one of the compositional type transforms.
So in this example here, I think it was like I personally try so many different transformation methods. Like when I have like a new data set, I just like follow the example and the kind of the transformation method that they had in the Bowman lab for kind of their data set. But yes, you want to for sure make sure that you look at different transformation methods and make sure that you're actually maintaining normality in your data.
And also, can you say anything about, in terms of correlating to outside measurements, continuous versus categorical data?
So if your end goal is to actually build a predictive model, then you wanna, and your system output is on a continuous scale. So for the sake of having quantitative output from your model, you are kind of forced to use Pearson rather than like Spearman, because it just like ranks, and that might not necessarily be something that you will be able to use or be useful to you to plug into other things like more complex model for ecosystem model, like a proper ecosystem model, rather than just like the model that you're building based on the regression. So you're just like compromising here.
Okay, so then that kind of gets back to Renee's question about the correlation method for building the network versus relating the modules to the external traits. Right, so the code you have, she was talking about line 153, the module trait correlation. But then on line 24, where you're building the network with the blockwise modules function, that also the default for that is Pearson correlation, but you could specify some other form of correlation.
Yes, I think you have control over that as well, Sharif. It's not going to be like a immediate plug and play, but there will be some like code messaging to get like an external correlation method inside this. But I think it's achievable to apply it within the like the function that builds the network as well.
Also, I guess the question,
yeah, like this two analyses are independent set of analyses. So yes, you have control over like we want to use this correlation methods here or there.
Yeah, so I guess the question then is, because I assume it would make sense since when you're constructing the network, you're correlating groups of genes to each other, essentially. And so then you would probably want to use potentially a more appropriate correlation method for the type of data you're utilizing. But when you're then at sort of the next higher level correlating, because when you're constructing the modules, or sorry, when you're doing the module trait correlation, you're actually, if I remember correctly, you're correlating the module eigengene, which is sort of like a centroid kind of to the trait. And so in that case, you're probably fine with the default bi-midweight or Pearson correlation. And that shouldn't be required. Or you shouldn't, in other words, that's a completely different type of correlation that you shouldn't necessarily need to go back to what the data is itself, right? Since you're just dealing with the module eigengene versus the trait you're looking at.
Yes, I completely agree with you on this, Sharif. So I have also another question here about temporal data. So temporal data are actually one of the tricky things here. If you are only looking at one dimension, so like a single experiment that is time 0, and then there are 2 other time points, you can still treat your time intervals as one of your environmental metadata, or one of the metadata, and then basically look at the change in the module composition across these different time points. That might be complicated if you also have different treatments. So you have time 0, and then time point 1 and 2, and treatment A, and then treatment B. Because basically time point 0 is basically the same thing shared between treatment 1 and treatment B, and the variation should happen later in the different time points. So in that case, I think you want to build different networks and then use the eigengenes to connect the modules from the different networks to each other. It will be more complicated, but this is, I guess, the way to go around it.
Could you actually follow up on that a little bit? Because you mentioned that earlier in the lecture, but using the eigengenes to correlate to other networks.
So basically, if you use the eigengenes from one module in one network and use the eigengenes from another module from another network, you can actually connect these modules to each other. So if these 2 modules are compositionally similar, so if they are the genes and the transcripts and the proteins of the same, the transcripts of the same genes and the proteins out of the same transcripts and proteins, that should be a strong signal for you and your data set, like about the importance of these genes and their expression signal to this ecosystem output. The example that I mentioned, and again, this would be like an ideal result to see, is, well, is there also differences between, if you look at the gene, the modules that are coming from the gene network versus the transcript network versus the protein network, and correlate all of these to the health versus disease phenotype. And you find that, well, yes, you have maybe like if we are using R-square as a metric here for the comparison, and the R-square is 0.6 at the gene level, it's 0.8 at the transcript level, and it's 0.9 at the protein level. This is something really informative from the biological perspective, that it is the existence of this metabolic pathway and the level of expression that controls the health outcome. So that's kind of like the example that I was thinking of. But I bet there are so many different applications as well of like doing this cross-network comparisons.
So that's more about relating the strength of connection to the external traits between each of the different networks than necessarily like somehow directly connecting modules constructed in different networks.
It can be also like this. I mean, you can still connect different modules from different networks. I'm not really seeing any more questions in the chat or the Q&A panel. Did I miss anything, Sharif?
No, everything is, yeah, there haven't been any additional questions.
So I don't want to keep people longer than this. Thank you so much for attending and for the chat and for the questions. And see you next week with Dylan and talking about mag construction from reads to genomes. Thank you so much again.